Anton's from Yale. He's been working for a long time on the RAS pathway and in particular on the effects of variants. And he's one of our superstars. So I'm super glad he's here. Anton, please.
Thank you so much, Tammy, for sharing that story. It's very heartfelt. I'm going to talk to you today about the basic science end of things, sort of trying to just dovetail from Tammy's message about the importance of research in this area. So I've always enjoyed coming to these meetings, and I'd like to thank the organizers for the invitation for me to speak today. So these are my disclosures. I'm a founder of Elargium Therapeutics, which is a phosphatase company that I founded. And some of the work that I'll be talking to you about today does cover some of the IP that Elargium Therapeutics is working on. So we've been working some time now on the protein tyrosine phosphatase, SHP2. Which is the protein tyrosine phosphatase that is encoded by the PTPN11 gene. SHP2 is ubiquitously expressed, and it functions downstream of multiple receptor tyrosine kinases to positively regulate activation of the MAP kinase pathway. But we and others have demonstrated that the activation of SHP2 itself plays an important role in engaging many other types of signaling pathways in addition to RAS, such as the calcium and the AKT signaling pathways, which are also very important in the actions of the phosphatase. SHP2 has been shown to be required for the development of growth, both during development as well as post-development. Now, as we've heard, we've been particularly interested in the variants of SHP2 as it's encoded by the PTPN11 gene, and multiple mutations in the PTPN11 gene can give rise to Noonan syndrome with multiple lentigines. And so we know that one of the major clinical phenotypes of NSML patients is hypertrophic cardiomyopathy. And we've been using mouse models of hypertrophic cardiomyopathy in knock-in mutants of the PTPN11 gene to understand the signaling pathways through which SHP2 signals, and subsequently what we can learn about those variants in terms of its downstream signaling. So one of the main things that I want to tell you about today is our perspective of how we think these PTPN11 variants are working. And fundamentally, we think that these mutants in the PTPN11 gene are essentially scaffolding mutations. So both PTPN11 mutations that lead to the activation of the phosphatase, as well as some mutations that lead to the inactivation of the phosphatase, as you know, cause overlapping types of phenotypes. But why is this? We are proposing that this is not related to the activity of the phosphatase itself, but rather these mutations cause an unfolding of the protein that lead to enhanced scaffolding features. Now, you can, in some instances, use a variety of small molecule inhibitors, as we know, to block the pathway. And some of these, such as the trametinib, as we know, have been very, very successful and are quite promising. However, not all of the PTPN11 associated mutations are going to be conducive to MEK inhibitors. So can we think about other types of inhibitors that are less stringent upon where SHP2 is acting in this pathway, and begin to think about those strategies as potential therapeutics? But in order to begin to identify what those therapeutics are, you have to understand the detailed molecular mechanisms of how these variants are actually driving signaling. And that's where my lab has been focusing on in terms of trying to understand this question. So we have shown through a number of studies that one of the major kinases that the PTPN11 associated mutations can interact with is a SRC family kinase. This is a tyrosine kinase that can phosphorylate a number of different upstream targets. And we've shown that there's an FDA-approved drug called dasatinib that can be used to ameliorate some, but not all, functions of PTPN11 associated symptoms in mouse models. And this is a slide that I added from a paper that we published several years ago, where we've used one of these NSML mouse models. This is the tyrosine 279-C mutant that was made by Maria Contreras several years ago. And in this mouse model, we treated mice with a low dose of dasatinib, and we followed them for several weeks. And we found that these mice, when treated with a low dose of dasatinib, was able to rescue the hypertrophic cardiomyopathy, as you can see from these echocardiographs. Now, as I've mentioned to you, we primarily use non-biased approaches in mouse models to try and identify differences in the signaling pathways that NSML-associated variants are driving as compared to wild type. I don't have time to go into all of the details of this pathway, but we've used phosphoproteomics and specific RNA sequencing approaches and couple those two omic types of technologies together to identify proteins which are altered in their levels of phosphorylation in the myocardium of hypertrophic cardiomyopathy cases in terms of where those pathways are in NSML versus wild type mice. And what we've identified is a protein called PZR. This is a protein that is phosphorylated on residues that tend to be phosphorylated by the kinase SRC, which, as I've just told you, is the target of dasatinib. So this protein is the most increased levels of phosphorylation in the hearts of these mice. And when you use dasatinib in these mice, the phosphorylation of this protein goes down, coincident with the restoration, the rescue of the hypertrophic cardiomyopathy. But that's correlative. What is the proof that phosphorylation of this protein is actually driving the decrease in the heart size? Is there a causative link? So I'm not going to go into all the details. This is what the protein looks like. We've shown that the protein binds to SHP2. So this protein, which is the most phosphorylated in the myocardium of NSML mice, also turns out to be a binding protein that interacts with SHP2. What does this mean? Well, it means that you can treat these mice with low-dose dasatinib to block the SRC kinase, and that rescues the hypertrophic cardiomyopathy. We've also done this genetically by making a mouse that does not become phosphorylated and hence does not interact with the phosphatase. And this is what happens. So these are wild-type mice that show the increased levels of phosphorylation in the NSML myocardium. And these mice are NSML mice that have been introduced with this mutant that does not become phosphorylated and it doesn't bind. Again, I don't have time to go into all the details, but this is a loss of function mutant. And this is what happens to the hearts. If you put this PZR mutant that does not become phosphorylated into the NSML mouse model, as you can see here, you can rescue the hypertrophic cardiomyopathy. So genetically and pharmacologically, we can rescue hypertrophic cardiomyopathy in the NSML background. So we're science geeks. What is the mechanism that is driving that? So in the next few minutes, I'll just tell you very briefly how we think this is working. As I mentioned, we used a lot of omic experiments, a lot of omic types of experiments. So this is an experiment where we've taken 4 genetic variants, performed RNA sequencing in the myocardium. You don't have to go into all the details of this heat map. But what I want to draw your attention to is the fact that we've identified a homeobox gene called the Iroquois 3 and 5, or abbreviated as IRX3 and IRX5, as target genes for the PZR protein. We've subsequently shown that these homeobox genes are involved in regulating many aspects of cardiac growth, both developmentally and post-developmentally. What's also very interesting is that IRX3 and IRX5 have been ascribed to cardiac repolarization effects. And as you know, NSML patients have been reported to have certain aspects of cardiac conduction disorders. So we are proposing that this PZR SHP2 signaling dysregulates these IRX3 and IRX5 homeobox genes in the myocardium of the NSML mouse model. We've gone on to show that these IRX3 and IRX5 proteins are increased in NSML mice as compared to their wild types. Importantly, you can take dasatinib and also restore IRX3 and IRX5 expression levels. You can do that genetically in the mouse model that I just explained, and you can also do it pharmacologically. Now, I mentioned to you that we believe that these NSML and other PTPN11 associated mutations are essentially a scaffolding defect where the phosphatase is normally closed in its wild type form, and then these mutations cause this destabilization of the phosphatase that then drives promiscuous signaling. And we've begun to look at a number of SHP2 associated mutants, and in all of those that we've tested, they drive activation of this IRX3, IRX5 pathway. Now, what we've found particularly interesting about this homeobox cassette is the fact that IRX3 and IRX5 target a factor called, I'm going to skip this actually, they signal rather to this SARC pathway, which then leads to the activation of this IRX3 and IRX5 signaling axis. But they all target the bone morphogenic protein 10. So when IRX3 and IRX5 get activated, they drive BMP10, and BMP10 has been shown to be involved in cardiogenesis. It's a cardiac-specific member of the TGF-beta family. And so that gives us an insight as to some of the cardiac selectivity of this pathway. BMP10 during development is expressed in the atrium, but has a significant amount of expression later on in life in certain parts of the myocardium. Mice that overexpress BMP10 have reduced heart size, suggesting that BMP10 negatively regulates cardiac growth. So what happens to BMP10 in the NSML mice? Well, it turns out that BMP10, through this PZR IRX3 IRX pathway, is suppressed in the NSML mice and is rescued when you put this PZR phosphorylation site mutant in, demonstrating that PZR phosphorylation, which interacts with SHP2, drives this IRX3, IRX5 signaling homeobox transcription factor cassette and suppresses BMP10. You can show that genetically, and very, very gratifyingly, you can take low-dose dasatinib and also significantly rescue the suppressed levels of BMP10 in these mice. So both BMP10 and low-dose dasatinib, which is the tyrosine kinase inhibitor, are working on the same axis. Now, since I've only got 5 minutes to wrap this up, I can tell you that we've gone on to show, and this is a paper that's coming out in PNAS, hopefully by the end of September, that you can recapitulate this entire pathway using human iPSC-derived cardiomyocytes. And so essentially you can take iPSC-derived cardiomyocytes and show that you can suppress signaling by putting in IRX3 and IRX5, and they work together to suppress the actions of BMP10 expression. Now, one of the things that we've been really trying to understand is I've told you that we believe that these PTPN11 associated mutants function as scaffolding, basically loss of scaffolding features, right? So you disrupt the folding of this protein. So what happens when you unfold the phosphatase with these mutants? So to do that, we had to turn to structural biology. And I'm not a structural biologist, but fortunately upstairs from me, there's some really very talented structural biologists who are happy to collaborate with me at Yale Medical School. And so what we did was we took our known interactors, mixed them together in a tube. They took them off to Brookhaven, and they've started working on various crystal structures to understand how these protein-protein interactions work. So I'm going to just show you a couple of pieces of data. It turns out that the unfolded conformation that we see with the PTPN11 associated mutants create a binding site for this kinase, the same kinase that turns out to be inhibited by dasatinib. That's more than a coincidence. We map the binding site, and in fact, I should show you that this binding site, which is mapped to the domain within the phosphatase, which is normally covered when the phosphatase is in its wild type configuration, is conserved amongst many, many species. Mice, xenopus, humans, flies have this motif. And this particular interaction is forced into an open conformation when you make the complex with this PZR. So what I'm showing you here is a co-crystal structure of these phosphorylated mutants bound to the SH2 domains of SHP2. And what that does is it keeps SHP2 in this open conformation and brings this kinase into close proximity. And so what dasatinib is doing, it's blocking the ability of the Sark family kinase to create additional PZR molecules that then assemble a very, very promiscuous signaling complex. And when you inhibit that, this complex, which we're showing now for the first time in 3D crystal structure, becomes non-functional. And if you do that in a disease context, that's sufficient to rescue the hypertrophic cardiomyopathy. So I've gone through a number of different, I'm going to actually skip this, but this is basically, you can read about this in the coming weeks, but I just want to leave you with one last slide here. If you take those proline-rich mutations, and one of these proline-rich mutations is actually an uncharacterized PTPN11 associated RASopathy mutation, by the way, that needs further investigation. But if you take these proline-rich mutants, you also completely block the pathway. So this interaction between this kinase and this aberrantly opened version of SHP2, we believe is a major driver of this promiscuous signaling that activates the AKT component of the pathway. So what I've told you today is that we have identified this homeobox cassette, IRX3 and IRX5, as an SHP2 associated NSML and NS mutant target gene. This pathway is activated in the hearts of NSML mice, and it targets BMP10. Really, what we think is going on here is this data supports the mechanism that these PTPN11 associated mutants are scaffolding related pathways, and that drives a potential pathway that is affecting heart growth. And the question is this the new BMP10 target that we've identified, does this really represent a new potential target for NSML as a biologic? But you know, this is highly speculative, of course, and we would love to continue that discussion. And with that, thank you very much for the invitation, and thanks for the attention.
Great. So we have time for a couple questions, if anybody has. Start here.
So what's next?
Where do you want to start? You know, so first of all, you know, we would love to, you know, do some trials with a low dose dasatinib. I've been coming to these meetings for, you know, a number of years now, and I think that we have a really good understanding as to how the dasatinib is working now at the molecular level, and we would love to move it in that direction. Please.
I'm going to skip over to here. Sorry, you did last time. Okay.
I'm going to be naughty. I'm going to ask a question I know the answer to because you told me at breakfast, but I thought it was really interesting, which is, given that dasatinib is used in leukemias, has got quite a high side effect profile in terms of muscle pains, fatigue, a lot of things that people with Noonan syndrome already suffer with. Can you tell us a little bit about the dose response that you're looking at in cardiomyopathy?
Did I give you the check after breakfast? And so no, that's a very, very good question. So one of the things that we've been able to do in dose response studies is that we've found that you can use an extremely low dose of the dasatinib when you look at the PZR as the endpoint target. And if you do the allometric calculations from humans down to mice, that dose is about 1 in 200th of that's used to treat leukemia. That's not to say that there won't be side effects treating patients, NSML patients, but the potential for those types of side effects, I think, would be significantly reduced at such a low dose. And we've shown that this is actually an on-target dose through pharmacokinetic experiments, specifically looking at the level of phosphorylation of PZR as a surrogate readout for the local ability of SARC to hit that target.
I'm going to take moderator's privilege and I'll
Go
ask you,
ahead.
I'm going to ask you the last question, so don't go away. So based on what you said, if I'd never met a patient with Noonan or NSML, I would expect their heart problems to be quite similar. But in fact, we know that in NSML, about 80% will have HCM, whereas in PTPN11 driven Noonan, especially the N3AD, it's like 6%. We almost never see it. So what are your insights into why the NSML alleles are resulting in HCM and the Noonan PTPN11 alleles are not?
So did you review my paper?
I did not.
Okay.
I did not.
So so in answer to Bruce's question, which was the same question that a reviewer asked, so we think that those sorts of differences are likely related to the KD effects, which are indirectly related to sort of the open versus the closed propensity of these various mutants. So in order to address that, we're going to need to do some more co-crystallography to understand how the openness of those mutations are affected in complex with those SH2 domains. Great question.
I swear I didn't review your paper, Anton. I'm an honest guy. You know that? I would say, yeah, it was me, but it wasn't. Okay, let's move on so we stay on time because he has to leave, of all things.